is an expert on redox biology and the kinetic modeling and metabolic control analysis. So Emma, please go ahead with your presentation.
Thank you very much. First of all, I want to thank to the organizers and especially to Ariel for inviting me to participate in this workshop. And here in this talk, I'm going to share our results on kinetic modeling and metabolic control analysis, and how we can we have used these two approaches to understand the control and regulation of metabolic pathways. Well, I'm going to take advantage of the previous speakers. Carlos already talked about the life cycle of Trypanosoma cruzi. I'm going to talk about that in this talk. Also, Pedro Mendez explained us about the capabilities of this wonderful tool, COPASI, for metabolic simulation. And Miguel explained us what is a special way of doing systems biology. In systems biology in general, the aim is to build computational models of complex cellular networks. And Miguel explained us the different types of models that can be built. The top-down strategy build this model using data derived from atomic studies. Usually these types of models discuss behavior patterns and cause-effect relationships of macromolecules. On the other hand, we have the bottom-up kinetic modeling, in which the models are built using the kinetic properties of the enzymes of the pathway reactions, and also as parameters of constraint thermodynamic parameters such as equilibrium constants. These models are more minimalistic, but they can give us information, and they help us to elucidate controlling and regulatory mechanisms. So here I'm going to give the examples of how what information can kinetic modeling give us. So what is needed to build a kinetic model of a metabolic pathway? Essentially, what we have to have the database of kinetic parameters of all the pathway reactions. What we have done to solve these problems, although there are a lot of kinetic parameters reported in the literature, they are usually very variable because the affinity constants for substrates or ligands, activators, inhibitors, usually they are determined on their optimal condition for the enzyme. But very often, these optimal conditions for the enzymes are not the physiological ones. So a key aspect of to obtain a good model is to determine these kinetic parameters under near physiological conditions, at least of a physiological pH. The cytosolic and mitochondrial physiological pH is between 7.2 or 7.4. And so we determine all the kinetic parameters using recombinant enzymes, or you can do it also by determining these parameters using cellular extracts. So with these kinetic parameters, we define the enzyme rate equation that includes the type of kinetic mechanisms reported in the literature for all the pathway enzymes, and use this marvelous COPASI software developed by Pedro Mendez and Ursula Kurmer, and who can perform a metabolic control analysis. And doing this iterative process of modeling, construction, and simulation, we use the metabolite concentration determined in live cells, and also the pathway fluxes determined in these cells under the same condition used here. So there is an iterative process. The first drafts of the model usually doesn't predict anything, but there is a model refining by going back to the experiment. So the kinetic model tells you what is the reaction that is not well modeled, and you go to the experiment looking for inhibitors to decrease the activity of one enzyme, or on the other side to increase the activity by determining the Ki, or the constant of activation of a particular reaction. So we perform this, and our type of models are of the ordinary differential equations that calculate the metabolite concentration through time to reach a steady state. So on the other hand, we have the metabolic control analysis, which is an approach that it is not so well known, but it is a very powerful framework to study the metabolic regulation and control regulation in metabolic pathways. This framework was developed in the early 70s by two independent groups in Scotland. Henry Catcher developed this theory, these concepts. He was a geneticist studying the molecular basis of gene dominance in human diseases. And on the other hand, the group of Reinhard Heinrich and Top Rappaport in East Germany, they were biophysicists. Well, Heinrich already passed away. Tom Rappaport is still working at Harvard. And they developed the first mathematical model of glycolysis in erythrocytes. So they reached the conclusion, working in these two different areas, that the control of a metabolic pathway was shared between all the pathway reactions. There was not a unique rate-limiting step or bottleneck reaction, as we read in the papers and biochemistry textbook. So they changed the way in looking how a metabolic pathway was controlled. So what we can do with metabolic control analysis? It allows to quantitatively determine the degree of control that each enzyme has on the pathway flux, which is called flux control coefficient, in which J is flux and AI is the activity of enzyme I. This also you can also quantitate the flux the concentration control coefficient, which has a similar meaning, but it is on the control that each enzyme has on the concentration of the pathway intermediates. So what is this flux control coefficient? What we have to do to determine this is to vary the enzyme activity individually without affecting all the pathway enzymes, all other pathway enzymes, and at each variation we have to determine the pathway flux, the changes in pathway flux. And in this hypothetical pathway of three enzymes, we have different behaviors. For example, as enzyme A, the flux control coefficient is the derivative at this point, the wild type enzyme level. And as you remember, to look at it visually, the derivative is the slope of the tangent to a specific point. So here we can see enzyme A, the slope is very high. It has a high control on flux, whereas we have another enzyme that basically it has no very low control on flux. And to inhibit the pathway flux, you need less inhibition of enzyme A, whereas enzyme C, you basically have to do a pharmacological knockout to affect the pathway flux. So there are some concepts derived in metabolic control analysis, as the flux control coefficient approaches to 1, means that the enzyme has the highest control of flux. The summation theorem of metabolic control analysis is that all the flux control coefficients have to add up 1. So as the enzyme has approaches to 1, it means it has a high control. And MCA studies have demonstrated that usually there are two or three main controlling steps. So in our field of parasitology, we have used this strategy to identify these controlling enzymes and to propose them as the most interesting therapeutic targets. So now, putting those backgrounds, I'm going to show you the results of kinetic modeling and MCA in 3 pathways: the trypanosoma metabolism, Trypanosoma cruzi, which is the causal agent of human Chagas disease, a very endemic and dangerous disease that afflicting many people in Latin America. Also glycolysis in Entamoeba histolytica, which is the causal agent of human amoebiasis. And I'm going to talk a little bit about cancer metabolism, glucose metabolism in cancer cells. So I'm going to start with the antioxidant metabolism in Trypanosoma cruzi. Trypanothione is the main antioxidant metabolite in these parasites. It is a conjugate of 2 glutathiones, which and bound by a spermidine molecule. And this replaces the function that glutathione has in our cell. Therefore, the trypanothione metabolism has been looked always as a good pathway for therapeutic intervention. And usually to determine the gene essentiality or enzyme essentiality, usually all the enzymes have been knocked out or knocked down, and it turns out that all are essential, and therefore everybody proposed the studied enzyme as putative drug targets. So we consider that additional criteria have to be applied for drug target prioritization, and we did so by identification of the main controlling steps by metabolic modeling and metabolic control analysis. So I'm not going to go in detail about each enzyme reaction. Every arrow here represents the kinetic parameters that we included in the model. So here, as I said, these are minimalistic models, but it gives us too much information. I hope to convince you of that. So the trypanothione synthesis is included these reactions. Trypanothione is synthesized from cysteine, glutamate, glycine, and spermidine by the action of gamma-glutamyl cysteine synthetase, glutathione synthetase, and trypanothione synthetase. And the spermidine is supplied from the extracellular milieu. In these models also included some regulatory kinetic inhibition of glutathione on trypanothione synthetase and glutathione on gamma-glutamyl cysteine synthetase, which are the most important regulatory loops in this pathway. And also we build this model in which of the peroxide reduction pathway, and including trypanothione reductase, tryparedoxin, and tryparedoxin peroxidase. So we defined each reaction with different types of kinetic mechanisms, random by substrate, random 3-uni, random or ordered type of reaction, ping pong, reversible, Haldane, mass action reaction, depending on the availability of the kinetic parameters. For example, for trypanothione demand, we don't have a specific reaction that define this. So usually this include this type of reaction as mass action reaction. So this is a sort of information that gives, we demonstrated using the recombinant enzyme that trypanothione synthetase was potently inhibited by glutathione, either in the recombinant enzyme or in parasites overexpressing this enzyme. And we find this type of equation. This is very simple, but we have very complicated other type of reactions, mechanisms. And this reaction included the mixed type inhibition by glutathione. And what COPASI can do or can provide us is the simulation. Here I simulated the changes, how these Ki values, constant of inhibition values, affect the homeostasis of the metabolic of the metabolites. For example, by increasing the inhibition of trypanosoma synthesis by glutathione, we can see that it mainly affects the glutathione pool, whereas in the by varying the Ki value of gamma glutamyl cysteine synthetase, it has homeostatic effect mainly on trypanothione, but not on glutathione, which is the main end pathway of the glutathione synthesis pathway. So it gives this type of information that we can simulate in the kinetic model. So after a process of refinement, trypanothione, are the simulations. This is the values determined in the parasites of metabolite concentration, and these are the model predictions. They are pretty close to the in vivo situation, and also the pathway flux predicted is within the same interval determined in the parasites. And this is the peroxide reduction simulation. The model can predict the ex vivo pathway flux. Here is within the same interval. So one advantage, as I said, and Pedro explained yesterday, COPASI is one of the softwares that can perform metabolic control analysis, and it provides this flux control coefficients, and these are the ones that were predicted by the model. Gamma glutamyl cysteine synthetase has the highest control on trypanothione, whereas trypanothione synthetase has lower control. We can rule glutathione synthetase, and the spermidine supply has a very, very low control of flux. On the other hand, in the peroxide reduction pathway, the reduction of tryparedoxine by trypanothione is the reaction or the process that has the highest control of the peroxide reduction pathway, whereas tryparedoxine is the next one in controlling, whereas trypanothione reductase has low control. Now this is interesting because many people around the world uses trypanothione reductase as an enzyme model for drug design studies. So someone can argue that, well, this is a predictions they obtained by modeling. Does it reflect the reality? What we did is to determine the flux control coefficients in vivo in the parasites. To do this, Xavi Gonzalez and Chitrali Vasquez obtained these plasmids that overexpress these different enzymes in the parasites, in the pemastegotes of the parasites. These parasites were transfected, and we obtained clones of this population that have different levels of enzyme activity. Even when you, in this process of transfection, the efficiency of transfection is not homogeneous. You have clones with different levels of activity or enzyme protein. So you have to clone to ensure that have populations that are homogeneous, at least for the protein of interest. And you have determined all the pathway enzymes to ensure that only the enzyme that you are modulating does not have effects on the other pathway enzymes. With these clones, the enzyme activities were determined to determine the metabolic changes in metabolic fluxes and determine the flux control coefficient. And this is the reality. This is the experiment, not very nicely to the simulation, to the model, to the model, but this is the experiment. This is the variation. But still, you can find interesting this is the flux control coefficient determined in vivo, here in this point of 100% of enzyme activity. As you can see, gamma ECS in vivo has the highest control, and this is the model, the value obtained by modeling. And by the summation theorem, trypanosine synthesis was predicted by the summation theorem. We did not have trypanosine synthesis changes in the clones because a modulation of this enzyme had modifications also in gamma ECS. So we had to rule out all, discard all these clones because there was unspecific effects of this transformation on other pathway enzymes. So the inhibition of gamma ECS and trypanosine synthesis could be the most, we decrease the trypanosine flux. In the peroxide reduction pathway, these are the experiments in the parasites. As you can see, tryparedoxin has again the highest control, and trypanosine reductase low control here. And these are the model, the flux control coefficients obtained by modeling. So there is a good match when you have a very controlled model, minimalistic, but well controlled. So these are the experiments. So inhibition of gamma ECS, trypanosine synthetase, and tryparedoxin will affect the antioxidant defense in cruzi. So is that real? So we use methionine sulfoxamine to, this is an allegedly specific inhibitor of gamma ECS. And parasites exposed to this increasing concentration of this methionine sulfoxamine, we observe indeed a decreasing glutathione. But despite having this 50% glutathione, we observe a very drastic depletion of trypanosine. So there was something wrong, and the model could tell us that BSO was not only inhibiting gamma ECS, but also trypanosine synthetase. And we tested this by using the recombinant enzyme and parasites that overexpress this trypanosine synthetase. And we confirmed that methionine sulfoxamine, this depletion of trypanosine was mainly due to inhibition of trypanosine synthetase. So also in our model, we asked why gamma ECS has such a high control on flux. So we thought that cysteine was a limiting metabolite for this pathway. So Ziplay performed an experiment in which she supplied parasites with increasing with cysteine. And indeed, she found that wild type parasites increase the trypanosine content threefold. And when she supplied parasites that overexpress gamma ECS, she obtained up to, with this dispersion, up to 8 times false trypanosine. So cysteine is a limiting metabolite for trypanosine synthesis and could account for the high control of gamma ECS on trypanosine synthesis. So now we embark on the studying the kinetic characterization of the cysteine supply ways to the parasites. And there are 3 different ways in which parasites can increase or modulate the cysteine pool, by increasing the transport of cysteine here, as here is observed here in this experiment. But the parasite also have the reverse transformation pathway, in which cysteine is synthesized from serine homocysteine by the action of cystathionine beta synthase as cystathionine gamma lyase, but also has this pathway present, this reverse transformation pathway. We have it in the mammalian cells, But the parasite also has the pathway present in plants that in which serine acetyltransferase and cysteine synthetase are using serine and acetyl coenzyme A produces cysteine. But the problem here in this pathway is that sulfide is very low in cells. It is so we don't know from where sulfide is coming in the parasite to account a flux for this one. So we are here in this point doing this characterization, and we included those reactions in the model. We already have the cysteine transport reaction and the reverse transsulfuration, and Javier Belmont characterized this reaction. He determined the pathway fluxes here and some metabolite concentration. We need still to verify the metabolite concentrations. And these enzymes have pinpoint kinetics. So these are the modeled the simulations of this a little bit amplified model, but the model can predict more or less the intermediary concentration. The pathway flux of trypanosoma synthesis did not change too much by adding these two reactions, and the model could predict the flux of cysteine synthesis in the parasites. So these are the flux control coefficients predicted by modeling. Still, the gamma glutamyl cysteine synthetase is the main control step of trypanosoma synthesis, whereas cysteine synthesis is mainly controlled by cystathionine gamma lyase, this enzyme, but also the control is shared by gamma glutamyl cysteine synthetase. So this enzyme is a keynote in the trypanosoma metabolism and cysteine synthesis. So we can get rid of the high control of gamma ECS. So these are modeled predictions, but Citlali Vasquez, what she did was to determine the flux control coefficient of cystathionine beta synthase, because we have the recombinant enzyme. In this experiment is very simple, but it also gives information. In a cellular extract, if you have the recombinant of pure enzyme, you can add different levels of activity and determine the pathway fluxes. And then you plot those data, and you can control that can determine the flux control coefficient. This is the obtained by this experiment, and this is the model value. So they are also very, very similar. So it is kinetic modeling is very powerful in that sense. So cystathionine gamma lyase is the enzyme that mainly controls the cysteine synthesis pathway in the parasite. So with this strategy now, from all the pathway reactions included or are considered in this model, we can now prioritize drug targets, cystathionine gamma lyase, gamma glutamyl cysteine synthetase, and tryparedoxin could be the most interesting therapeutic targets. Trypanosoma synthetase, although it has low control, without this enzyme, no trypanosoma is synthesized, and it affects all the antioxidant metabolism in the parasite. And trypanosoma reductase, as I already said, basically every week, paper on finding specific and very potent inhibitors of trypanosoma reductase are published, but it is a very difficult drug target because it has low control in the pathway. And this is because you have the parasite has a high activity, or it is until 100 times the fluxes in this. The parasite has 100 times higher activity of trypanosoma reductase that surpasses the peroxide reduction flux. So you have an excess, the parasite has an excess of trypanosoma reductase. That's why it is a very difficult drug target. Now I'm going to talk about kinetic modeling of Entamoeba histolytica glycolysis. I'm going to illustrate this of PPI-dependent glycolysis because it is a very different pathway of that found in cells. This is a pathway that doesn't have the ATP phosphofructokinase and pyruvate kinase, and this reaction uses pyrophosphate instead of ATP or ADP. So this the question here in this pathway was from where the pyrophosphate was coming, and it was there were other reactions that provide pyrophosphate in the anabolism here in the glycogen cycling, but the inclusion of this reaction didn't predict well the pathway fluxes, and therefore it was necessary to include other reactions. So here I show you that the glycogen cycling is a way described in other PPI-dependent glycolysis from other organisms that provides pyrophosphate. And just by including this reaction, the model prediction, the model worked. So it was not possible discovering this by just imagining. The prediction was done by kinetic modeling. And just to summarize here, these are the modeled flux control coefficients obtained by model, and by applying other experimental strategies of metabolic control analysis, we could obtain the pathway, the flux control coefficients by titration with inhibitors and elasticity analysis in live cells, which does not imply too much genetic manipulation in the parasites. So it is very powerful. And here I show you the what could happen in the parasites when you inhibit high or low controlling enzymes. 90% inhibition of a low controlling enzyme such as P4 has no effect on the ATP content or cell viability. But if you inhibit a high controlling step here in these pathways, here, you indeed decrease the ATP content 50%, but does not affect too much the cell viability. Whereas if you inhibit the two main controlling steps, you now observe the effect on cell variability. So this plot also tells you that to affect the cell viability, you have to decrease the ATP content by more than 50%, not 10% or 20%. You have to really decrease the ATP content to affect the cell viability. So I'm not going to talk about cancer cells. So the general conclusion is that kinetic modeling allows understanding of regulatory and controlling mechanisms, and they allow to propose drug target candidates. This is my group, several students participating in different occasions, the grants by the CONACYT Mexico financial agent, and my collaborators, Dr. Rafael Moreno Sanchez and Dr. Marin Hernandez, who has working on metabolic and kinetic modeling of glucose metabolism in cancer cells. So that's all from my side. Thank you.
So thank you very much, Emma, for this fantastic talk.